All right, it's my pleasure to introduce our first speaker, Eva. She's joining us from the University of California, Davis, where she's currently a PhD candidate. Her passion is the study of viruses that affect fish and the application of research for the improvement of clinical medicine of aquatic species. So Eva, turn it over to you.
Cool. Thanks, Nick. Hi, everyone. As Nick just said, my name is Quijano Cardé. I'm a PhD candidate at the University of California, Davis, under the guidance of Esteban Soto, and I'm very excited today to present an update on two projects that highlight the usefulness of two different types of DNA vectors in improving the detection and the study of DNA viruses in fish. Just want to take a second to acknowledge my collaborators on this project. It has been very fun to work with them, and I'm very excited to share this. Okay, so I just want to start by saying that I love viruses, and I know that there's a few of us out there that love viruses as well in this group. But if we look at aquatic microbiology as a whole, fish viruses are understudied in comparison to fish bacteria, for example. And the reason is multifactorial, but includes the need for additional cell lines to grow them, the lack of certain validated molecular techniques to study them, and the need for more experts focused on the field of fish virology. What we don't lack is viruses to study. So this is a list of viruses in aquaculture. I don't want you to focus on reading all of these abbreviations. It's just to illustrate that this is just a small list of significant viruses out there requiring more research. And some of them, which I have bolded, are incredibly important due to their effects on the target host populations. And therefore, they have been even listed as notifiable by the World Organization of Animal Health. In addition, we can think about the potential for latency in the Alloherpes viridae family and how that is an aspect of these viruses that we know very little about, despite how important it has been shown to be in other families of the order Herpes viralis, most notably those that affect humans or major pet species like the cat. And finally, I want to say that there are other viruses in this group, like salmon herpes virus 4 and carpedema virus, that haven't been successfully cultured in vitro, despite their importance in the species they affect. So the two projects that I will describe today are a reflection of my desire to know more about these viruses and therefore develop and validate molecular tools that allow us to study them and focus on their pathogenesis. And we will focus on those two issues that I just highlighted, latency and unculturable viruses. So let's start by talking a little bit about DNA vectors for those of you that don't have a lot of experience with them. These are particles used as a vehicle to carry any foreign DNA into a cell where it can be replicated or expressed. And there's a few types of DNA vectors that are very well known in molecular biology. We have plasmids, which we'll talk in just a minute about. Viruses, which phages are probably the most common that people hear about. Artificial chromosomes, which can be bacterial, yeast, or human in origin, and we'll discuss those during the second half of this talk. And cosmids, which are kind of like a hybrid between a plasmid and a lambda phage vector, and they're capable of cloning large segments of DNA. What they all have in common at the very minimum is an origin of replication, a multi-cloning site, and a selectable marker. So the first talk, for the first half of this talk, we're going to focus on plasmids. And these are extrachromosomal, circular, double-stranded DNA molecules that are self-replicating within the host. And they easily generate numerous copies for our use. And despite making a significant number of copies, in general, plasmids are pretty stable, which is a great thing. The one drawback about plasmids is the small insert size of up to only 15,000 base pairs. One awesome thing about engineered plasmids that we're going to exploit in this project is their multiple cloning site, an area with multiple restriction sites for easy insertion and subsequent transfer of a gene of interest into a target cell. And so how can we use this to improve detection and study of DNA viruses in fish? So I'm going to answer your question with another question. Why are some viruses unculturable? So there are numerous reasons why a virus will not grow in cell culture, ranging from low levels of pathogen in the sample, poor growth conditions, the presence of growth suppressive factors in the actual specimen that we're inoculating onto those cells, the lack of entry receptors in the cell line that's available, and rapid immune clearance by the cell that's being infected. And these are just examples. It's not all the reasons why a virus may not grow. And while tackling all of these factors is quite the undertaking, maybe modifying just one could make all the difference in a virus growth. And the one that we decided to look at is immune clearance. So the immune response to viruses is very complex and involves a myriad of molecules. But one important protein in the response against viruses is known as protein kinase R, which we'll call PKR. And PKR is constitutively expressed and accumulated in the nucleus and cytoplasm as an inactive monomer. And so we have that little depiction here on the right. So when this inactive monomer is activated mainly by the presence of double-stranded RNA, which is generated during viral infection, PKR phosphorylates the alpha subunit of the eukaryotic translational initiation factor 2, which is called EIF2 alpha down here in this depiction. This phosphorylation prevents the subsequent use of EIF2, leading to the inhibition of protein synthesis and therefore halting of the viral protein production. If this wasn't enough, PKR has also been described to be involved in apoptosis and in the amplification of the interferon response, which we all know is crucial in the antiviral response. And because of these critical roles in antiviral response, you can imagine that the PKR pathway is actually a common target for viruses. And if you imagine that, you would be right. In fact, vaccinia virus, which is a member of the family Poxviridae, has two well-described PKR inhibitors, E3L, which is in this little picture, and K3L. E3L in particular is an overachiever because it is not only a direct inhibitor of PKR via protein-protein interactions, but it also plays a significant role by sequestering double-stranded RNA molecules from the virus. So at this point, you may suspect where I'm going with this. So let's say we have a fish, a carp to be more specific, and this particular image was made by Dr. Taylor Heckman, who is on this meeting. So when we look at the genome of the carp, we find that the PKR coding sequence is present. So potentially we can imagine that cell lines originating from the carp could be considerably expressing and accumulating PKR inactive monomers. And let's say we have a virus, carpedema virus, to be more precise. The genome of carpedema virus, also known as CEV, lacks any known PKR inhibitors. And guess what? Which you know because I just told you. Currently, we have been unsuccessful in culturing CEV in vitro using any of the available fish cell lines. So what if we use a plasmid that contained the E3L gene and transfected a target cell for CEV? If the carp PKR is playing a significant role in preventing the growth of CEV in vitro, then by using a PKR inhibitor, we may just get CEV to grow. And our collaborator, Dr. Stefan Rothenberg, has used this strategy before to grow other pox viruses in vitro that are a little fastidious to grow, like myxoma virus in rabbits. So before we move forward, let me step back and speak about our patient and its virus for those of you that haven't worked with carp or CEV before. So carp are important aquaculture species worldwide, being one of the primary freshwater fish species cultured in the year 2020. And for reference, in the United States, 462 tons of carp were sold, generating about $2.3 million in revenue during that year. And an important pathogen of carp is carpedema virus, or CEV. And this is a double-stranded DNA virus in the family Poxviridae that is capable of infecting a variety of carp species, including the ornamental variety, the koi, which is actually what it's depicted on this image. And the koi has been diagnosed with this virus across the United States since 1996. But in addition to its prevalence in the ornamental industry, CEV has been associated with wild fish mortality events in the Midwest of the United States, and it's now actually reported as an emerging pathogen by the World Organization for Animal Health. And while the severity of clinical signs varies between carp strains, CEV usually causes lethargy, anorexia, enophthalmos, pale swollen gills, and somewhere between 75 and 100% mortality in juvenile fish. And the syndrome seen during advanced stages of this disease is called sleepy koi disease, which is what people usually have heard about. And this is because fish are typically found at the bottom of the pond in lateral recumbency, and they're only awakened when you disturb the tank or the pond. Okay, so let's get back to the experiment. To look into whether the expression of a PKR inhibitor by coefin cells, which are also known as KF1 cells down here, will allow the propagation of CV in vitro, we have been using a plasmid that contains a EGFP E3L fusion gene made by cloning the viral E3L gene from vaccinia virus into this PEGFP N1 vector, which is a vector that you can find in any local store. And this fusion protein will be under the control of the cytomegalovirus promoter. And I hope you can see my cursor, but it's right here, that promoter. This particular engineer plasmid was actually designed by Dr. Stefan Ruthenberg and his team, one of our collaborators. And briefly, I took this plasmid and transfected these KF1 cells using the GenJet DNA in vitro transfection agent, which is a type of transfection agent that carries the plasmid to be delivered within these particles of covalently cross-linked cationic lipids with polymer. And then the cells that were successfully transfected, I selected for pure culture using the G418 geneticin selective antibiotic, since the plasmid has a neomycin-resistant cassette right here. And of course, I did a geneticin kill curve of the cells that I will show you in a minute to determine the minimum concentration that kills the susceptible cells. And monoculture of these cells have been growing from a small pool of selected cells. And I'll show you the assessment of the EGFP signal at the end. Okay, so this is the geneticin kill curve. So we have KF1 cells here plated in T25 flasks, and this is a 2-week curve. So these images were taken at 14 days post-geneticin. On the left, we have the negative control, which just received normal growth media, MEM, supplemented with FBS. And we can see that that's a nice monoculture of KF1 cells. And when we add increasing concentration of the geneticin in micrograms per milliliter, we can see that at 100 and 300, we have significant rounding of the cells. And at 300, we even have some lifting of those cells, reflecting toxicity and stress. By the 500 microgram per mil solution, we can see that most of the cells have lifted and are dead at this point, reflecting a high toxicity of the cells at this concentration. And 700, 900, pretty much the same thing. All the cells are dead. But because of 500, all the cells were dead already, we're going to use that as the concentration, the minimum concentration that will kill sensitive cells to geneticin. So here we have the fluorescence assessment of these transfected cell lines. This is pretty exciting to me. It had been done with 2 other fish cell lines before, but never with KF1s or sturgeon cells, which I'm also transfecting, but I'm not going to talk about those right now. So on the left, we have the white light, the normal microscopy light. So we have here a little KF1 cell in the middle, and we also have a couple of like rounded cells surrounding them, reflecting that they're not doing well. And then these images are like 10 days post-geneticin, so about 14 days post-transfection. And on the right, we can see a really strong GFP signal on this KF1 cell cytoplasm and the little nucleus in the middle that has no signal, which is expected. And then this is pretty much the same. I just wanted to show you this because we can see that at 10 days, we have a cluster of cells here and some cells at the bottom that are still alive, but I expect these cells to die because we can see on the fluorescence window that only one cell in that cluster has GFP signal. So I expect that cell to be the only one that's going to be resistant to geneticin. So right now I'm still growing the new transfected cell lines from the individual cells that survived the geneticin killing. And once I have this cell line propagated, I'm going to infect it with CEV infected tissues that we've collected from clinical cases here in California. So stay tuned for that. If we are successful, this strategy could also be employed in other similar cases of fish host and virus, not only for other unculturable viruses, but to grow slow-growing viruses more quickly and as a sensitive cell line when screening populations for viral disease, which we do a lot in the aquaculture industry. Okay, so let's switch gears a little bit, but not that far away from the plasmids, to a different type of vector called a bacterial artificial chromosome, which we also know as a BAC. And as you can see from this map on the right, BACs look a lot like plasmids, but they have a handful of special features and a huge advantage. So the first thing is that a BAC has an origin replication of the fertility factor in E. coli, which allows control of low copy number per bacterial cell. So we're talking like 1 to 2 copies per E. coli. And while a low copy number can be annoying when it comes to generating enough BACs for us to use in the lab, the low copy number results in reducing the metabolic burden imposed on the host cell, and it makes the BAC extremely stable and is way less likely to accumulate mutations. However, having a low copy number prevents them from relying on diffusion for their distribution to daughter cells. So to solve this problem, the second special feature of a BAC is a partition mechanism right here, which acts resembling the mitosis process of eukaryotic cells and ensures proper distribution of plasmid molecules to all daughter cells. The biggest advantage of BACs to us over plasmid is their capability to have inserts of up to 300,000 base pairs. And some of the yeast artificial chromosomes can actually hold up to 500 kb. So with an insert capacity of 300 kb, that means that BACs can hold complete viral genomes of double-stranded DNA viruses. And that's a huge deal for us for one main reason. It makes it way easier to produce recombinant viral strains. And you can actually do it inside E. coli, which we'll talk about in a second. So if you know me, you know that at some point, I'm going to talk about herpes, which is my favorite type of virus. And our herpes viruses, the family of herpes viruses that infects fish and amphibians, have pretty large genomes, roughly between 120 and 250 kb, making them pretty poor candidates for insertion into a plasmid. But they have a particular part of their life cycle that makes them excellent candidates for the construction of viral BACs, and that is the circularization in the nucleus during replication. And I'll talk about why that is important in just a second. But one final advantage of building viral BACs is for storage and maintenance of the virus in the absence of fish or amphibians that host this virus in the lab. So due to their low copy number and stability, maintenance in the form of a viral BAC will lower the risk of becoming attenuated by passaging in vitro culture. So here's a little depiction of, in general, the development of a viral BAC. This example uses human cytomegalovirus, but the concept applies to other herpes viruses as well. And I'll show you what we have done in our project in just a minute. So remember how I told you that circularization of the virus during replication was advantageous? This is because we can exploit that to make it easier to generate the viral BAC. So we can transfect the BAC vector right here with homology arms to the virus genome, and we can infect at the same time with the virus. And during the infection process, the herpes viral DNA will circularize and the homologous recombination will insert the BAC vector into the viral DNA. And from there, we can identify infectious particles that picked up the BAC by assessing fluorescence of viral plaques in the cell culture. And of course, after that, as we mentioned, we can isolate the viral BAC, store it, mutate it within E. coli mutagenesis systems, and then obtain new infectious mutants, which is pretty cool. So let me quickly tell you about our virus of interest and how we're using a BAC vector to study its pathogenesis. So acid-sensory herpes virus 2, also known as AFS-FHS, is a large double-stranded DNA virus in the family Alloherpesviridae that affects mostly juvenile and subadult white sturgeon, causing epidermal ulceration like these ones seen on the screen, inappetence, lethargy, erratic swimming, and between 50 and 80% mortality in natural outbreaks. And this is very significant considering that in the United States, white sturgeon is responsible for roughly 95% of the caviar and sturgeon meat production, which generates over $200 million in yearly revenue. A particularly interesting aspect of herpes viruses, as I mentioned before, is latent escapability. And as far as I know, we have not been able to describe the latent state of AFS-FHS2, including the cell tropism during this life stage. So what we have done is taken this commercial P-BELO11 vector and cloned this fragment using the SAL1 restriction enzyme. And this first modification to the P-BELO11 will give the VAC vector hygromycin resistant due to this antimicrobial resistant cassette, and an EGFP fluorescence constitutively expressed due to the control of this elongation factor 1 alpha promoter. And we then cloned the 2 homology arms to the ACHV2 genome using the HPA1 site to be able to form the viral back by homologous recombination. And I'll show you where the homologous arms are located within the genome of the virus in just a second. We also inserted this LOXP system in the site to be able to excise the back vector surrounding the viral genome if we desire after we mutate the virus. So up here is the ACHV2 genome, which will be published very soon. And we selected this space right here between ORFs 21 and 22 because they're facing each other. So the insertion of the back won't disrupt any coding sequence or any poly A sites that are important for that process. And these down here are the sequencing of those homology arms, the left and the right. And I've aligned them to the sequence of UCD330, which is a strain of ACHV2. And I just want to show you that we got really good sequence fidelity with only like 3 sites that are different from the sequence that we expected. And insertion of the back would happen right here between those ORFs that I mentioned with no disruption to them at all. So where am I right now with this project? I have a back vector made for homologous recombination with ACHV2. And I have at this time infected WSSK cells, which are from white sturgeon skin, and have transfected them with the back vector. So I'm waiting for some CPE. Today is actually day 7 post-infection. So cross your fingers for me. I'm gonna check for CPE and hopefully we'll have some of that I'll be able to assess for EGFP positive plaques very soon. While I get that viral back done, I also got these neo-hatchlings from white sturgeon. They look very adorable. They're growing up to be infected in the near future with one of these mutant viruses. So we want to infect them with a fluorescent tag virus to be able to track the virus throughout the infection process. So stay tuned to that. If we are successful, we will have a more complete idea of the temporal viral distribution within the host, and we'll be able to use it for other viruses. So quickly, just want to acknowledge all the people that really helped me with this project and are still helping me in the process of it. To the Aquatic Animal Health Lab here on the right, especially Dr. Esteban Soto with all his guidance and encouragement, and to all my peeps that are always willing to help me with experiments and go out to eat with me when I'm feeling very sad about experiments that fail. And to my collaborators, Dr. Yoshihiro Itsumiya and Dr. Stefan Rothenberg and his team, especially students Adra and Joanna at the Itsumiya Lab, and Erin and Dewi for all the help with fluorescent assessment. And then to my funding sources and the laboratories that are involved. Thank you so much.
Excellent presentation, very interesting work.
Thank you.
We've got a few minutes for questions if folks want to either unmute themselves or drop them in the chat.
Hi, Eva.
Hey.
Mikola here. So you are juggling 2 big projects. How you are coping with it? Because I'm more interested in your results with CEV, and I don't care about the herpes virus.
That's fair. That's fair.
And I have a question about the CEV. So you say that PKR is so important in responses to the virus. Wouldn't it be easier to just CRISPR-Cas this out of the cells and get the replication in this way? So,
I'm not sure if it would be easier. The plasmid is pretty easy to insert, to transfect into mammalian cells. That's why we wanted to try that first, because it has been done on channel catfish ovary cells and also fathead minnow cells. So I knew that it was possible to transfect it into fish cells. And that's why we decided to do this first. That's a great idea to try if it ends up happening that the either like the plasmid expression of the actual PKR inhibitor is not great in the fish cell line, but we see maybe like a tiny little bit of influence in generation of growth. If that
I
makes
have
sense.
one more. Yeah, great answer. I have one more question, if I may.
Yeah.
So you are using E3L, which is pox virus protein from mammals and higher
from
mammals.
virus. Mm-hmm.
So do you think that this will work for fish? One thing, and secondly, were you considering using some proteins which are dismantling immune responses in fish? So, for example, proteins from Cyprian herpes virus 3, like ORF112, which was shown to be a very strong inhibitor of type 1 interferon responses.
Yeah, the reason why we picked the E3L is mainly because there have been studies looking at the ability of E3L to impact growth of other viruses that are not vaccinia virus. So we know it has a broad activity. I don't have evidence that it will be particularly effective against CEV or fish viruses, but I think it'll be interesting to see. I think what would also be interesting is grabbing if there are any PKR inhibitor homologs from other fish viruses and trying that. But because they haven't been characterized, I decided to start with E3L, which has been studied extensively with vaccinia.
Yeah, so ORF112 is an inhibitor of PKZ. So this is a special fish protein, which is having probably a bigger impact for the DNA viruses than PKR itself, although the E3L might be also inhibitor of this one. We don't know. I'm just afraid that you are slightly far away from the host. Yeah, so with the protein, this might not be fitting to interact with fish PKR. But keep on doing, and you're doing a great job.
Thank you. No, and that's a really good idea. If it's an inhibitor of PKZ, maybe it has some cross activity with PKR. So that would be good to try.
Awesome, great questions. For time, we're going to have to end it there. Thank you very much, Eva.
Yeah, thank you. I'll answer Melina's question on the chat.
Perfect. Thank you.